Hello, everybody. Welcome to this fortnight's breast tumour grand rounds. Just to begin with, I would like to acknowledge the traditional custodians of the land on which myself and today's presenters and the audience are located. I'd like to pay respects to the elders, both past and present, and extend that respect to other Indigenous Australians who may be present online. So there are a few housekeeping notes to take care of before we move forward. So as you would know, today's event is being recorded and will be available on the VCCC Alliance Open Learning page, where you can also view some of our previous webinars. If you have any questions for today's presenters, you can do this via the Zoom Q&A function. So pop your questions in there and I can read them out to Kylie for you at the end. And we encourage you to submit your questions throughout the presentation, and then we will get to the questions at the end of the session. So it gives me a really great pleasure to introduce our speaker for today, who is a good friend and just an awesome person, Associate Professor Kylie Gorringe. She undertook her PhD at Cambridge University, and this was followed by postdoctoral positions at the Peter MacCallum Cancer Centre and also at James Cook University. She held the NHMRC Career Development and VCA Mid-Career Fellowships and has been a group leader at Peter Mac since 2016. So her group's research really focuses on using genomics to address clinical questions in breast and ovarian cancer. And they're developing molecular prognostic models for pre-invasive breast lesions, including ductal carcinoma in situ and antidiuretic hormone. I think you might mean ADH, which is atypical ductal hyperplasia. And they're studying rare ovarian cancer subtypes and developing novel preclinical models for testing therapies. Her work has been funded by grants from NHMRC, NBCF, and the US Department of Defense. And as for those people who know her around the precinct, she's also a really strong advocate for women in science. So we look forward to hearing what you're going to say, Kylie.
Thanks so much, Cara. That was a really kind introduction. I will just share my presentation. And where are we? So hopefully you can now. Is that the right one? Now I'm thinking it's the wrong one. It's good? Perfect. Thanks, Cara. So thanks very much, everyone, for coming to listen to me talk today. As Cara said, my group works on a couple of different areas, but I'm going to be focusing on breast cancer today. So we're really interested in our studies of breast cancer on those stages leading up to the invasive breast cancer that affects so many women. And particularly interested in developing biomarkers because these pre-invasive lesions are actually clinically, a fairly important problem. So with atypical ductal hyperplasia, we know that there's a risk of upgrade, so carcinoma occurring in the same breast when they're diagnosed on biopsy, and also that they can progress to a more aggressive form of cancer. With DCIS, again, this is a big clinical problem in diagnosis of DCIS and overtreatment. We want to understand not only what biomarkers we can use to predict the development of invasive breast cancer, but also that process of becoming invasive, which still remains a mystery. So in terms of the team that have been working on this with me, I've just got some pictures of some of my key lab people. Also a shout out to David Byrne, who's doesn't work for me, but works for Stephen Fox, but has been really helpful with all of these projects I'm going to talk about today. And of course, you can't talk about this sort of work without mentioning collaborators. And these are some of my key collaborators who've been instrumental in helping obtain the tissues that we study, and also for their fantastic intellectual input also. So current projects that are going on in the lab at the moment, as I said, interested in biomarkers. So the first one is validating a biomarker for prediction of upgrade after what's called a B3 diagnosis, such as ADH. This is work now just been funded by Cancer Australia in partnership with NBCF. And Tangine is working on this, developing a copy number assay using FISH and also looking at the lymphocytes. Our second one, which has been going on for a long time, which is really about DCIS recurrence, funded by NHMRC and Victorian Cancer Agency, looking at a number of different modalities. But I've talked about both of those projects in the past, so I'm not going to mention them much other than in passing. And what I really want to focus on today is the understanding of breast cancer progression and evolution. And this is a study that's almost completed now, that was funded by NBCF. So when we think about cancer as a process, It's an evolutionary process, an evolutionary development. And the figure that I'm showing you here is from this fantastic study, which is almost as old as I am, by Peter Now. And he was the first person that really kind of put it together, this concept of an evolutionary process of cancer, whereby we start with some kind of initiating event, almost like a genetic event. And then all of the subsequent clonal expansion, so cells that grow from that, selection and competition is this evolutionary process where we grow out multiple cells, multiple sort of branching until we end up with what we see in the clinic of a clonal expansion that are all derived from this initial ancestral cells. And so I'm going to be using the word clone quite a lot and clonal throughout this talk. And that's really what I mean, that all of these cancer cells that we see at the end are clonally related. They've all come from this initiating cell and all share that initiating genetic events. And of course, as they develop subclones. And so we can have multiple subclones that are more or less related when we look at the final tumor and as we see that progression. So the types of genetic events, of course, that we can see include point mutations. These might be cause loss of function or gain of function in the sense of oncogenes. And most importantly, I think for breast cancer, although they do have point mutations, they also have a lot of chromosome changes. So we might have copy number events. For example, there might be losses, there might be increases, copy number gains. These can be of short bits or whole chromosomes even. And then, of course, there can be these structural rearrangements, insertions, translocations, and high-level gain. And so we can see these three karyotypes here are all from breast cancer cell lines. And we can see how that we can have ones that are quite stable, that are diploid, maybe have one or two changes. But then many breast cancers go through this process of whole genome duplication. And they are, you can see how deranged this genome is. There's multiple translocations. It's highly aneuploid. And you can tell that it's gone through this whole genome duplication because you have double copies of these quite complex translocations. So this would have happened while the cell's still diploid, and then everything's been duplicated. And after that duplication, there's been additional things happening. And that's this kind of evolutionary process that we're trying to understand in more detail. So obviously when we want to detect mutations and copy number change and structural variants, how we do it these days is, of course, by sequencing. Now, the most powerful approach is whole genome sequencing, where you're looking pretty much at everything. Your library contains everything. You can also do whole exome sequencing, where you're just looking at the coding regions of the genome. This is a slightly more efficient way to get to those, those real point mutations. And as well, you can also do targeted sequencing, which historically was considerably cheaper than these other two, although that distinction is not so strong anymore. But where you're just looking at a subset of the genes and usually just looking at coding regions again. And we can use these libraries to identify point mutations. So for example, here's some reads, all the grey lines are individual DNA reads, and you can see the change where the reference base C has changed to a T. So we can pick up these mutations in the sequencing reads. But we can also pick up copy number events as well. And you can see that here where there's a deletion, and that means there are fewer reads compared to the surrounding regions. And of course, if we have whole genome sequencing, we can also get the breakpoints of those structural variations and start to piece those together as well. So most of what I'm going to talk about today will be either using whole exome sequencing or targeted sequencing. All right, so what about breast cancer progression? So similar to what I mentioned before, this kicks off with some kind of event in an initiating cell. And in some cases, we have some ideas about what that initiating cell is, and it may vary. So the type of cell that initiating event happens in could determine whether you go down an ER negative, ER positive, HER2. Different types of breast cancer may start in different initiating cells. But effectively, whatever the event is, it leads to an increase in cell number, some kind of increase in proliferation, and you get this hyperplasia. Further development means the cells start to look funny, they look atypical, and we can get this with an atypical ductal hyperplasia. And this is the ones where we would sort of say this is like this B3 lesion that are problematic for diagnosis. Then from there, we can go on to a ductal carcinoma in situ, where now they're proliferating enough that they're nearly filling the duct, and they really start to look like cancer cells. And finally, of course, going through to the invasive when they escape out of the duct into the surrounding tissue. So we're particularly interested in these particular phases here. What does it take to go from an ADH to a DCIS? Are there genetic changes involved? are the genetic changes to get from DCIS to invasive cancer. And keeping in mind, of course, that this is a fairly simplistic model, and there are the complexities of grade, high and low grade, ER positive, ER negative, HER2 status, etc., that we need to take into account in the analysis. So we started off thinking about ADH. We've done some work with ADH. This was work done by Tangino, who was a PhD student in the lab at the time. And we're looking at copy number, and we knew that copy number was important, and you can indeed see these copy number changes at this ADH stage. So here what I'm showing you, this is just a percentage of samples that have copy number change at a particular locus in the chromosome, from chromosome 1 to chromosome X, gains in blue, losses in red. And so you can see out of the ADH, most of them had a copy number change, but it was fairly in just a few locations that you can see here. Now, in comparison, when we looked at DCIS, so this is pure DCIS, not seen with anything else in the same breast, you can see how many more changes there are. Now, some of these are the same as the ones we see in ADH, as you can see suggested here. But of course, we get some additional ones that we really don't see in ADH very often at all, but we do see in DCIS, and we do see in invasive cancer. And we can quantify this by counting the amount of the genome that's affected by copy number change, and we see a very strong increase in copy number events between ADH and DCIS, and a little bit additional copy number changes between DCIS and invasive cancer, although not quite as striking. But that was cases that were independent of each other. We've taken a particular tumour at a particular point of time and just looked at the one thing that was there. There was an ADH there, or there was a DCIS, or there was an invasive cancer. But what do we want to understand the tumour development within the same person? So what happened to that ADH when it became DCIS? So one way that we can do this is by looking at lesions that exist together. So this happens more often than you might think, that you have in the same breast at the same time, you have multiple lesions. So here's an example where you've got an area of invasive cancer, an area of DCIS, and a distant area of ADH. And what we can do is we can carefully dissect out the cells from each of those regions, extract the DNA, and then look at their genomic profiles and compare. And what we can look for is if we see that there are shared genetic events, again, these might have happened in that initiating cell or somewhere early along the way, we can say that they've had a common ancestral cell and they are clonal, they're related to each other. If we don't see any shared events, then we can infer that possibly they might be of an independent origin. So when we did this with ADH, with coexisting carcinomas, indeed the majority of them showed a clonal relationship to the carcinoma that was present in the same breast at the same time. And this didn't matter whether the carcinoma was low or high grade, or even if it was ER positive or ER negative. We often saw a relationship between the ADH and the coexisting carcinoma, suggesting that they were derived, they were on that same clonal pathway, and maybe the ADH may have been developed into the cancer. And when we look at these in individual cases, so what I'm showing here is now it's not a percentage, these are now individual profiles. So this is one particular ADH sample that was found with a DCIS and an invasive cancer. And similarly, here's another pair, ADH invasive, ADH and DCIS. Losses in red, gains in blue. And so we can see that clonal relationship in this copy number data where they share particular events. But we also see as they progress, they often gain additional copy number changes. As you can see here, this DCIS has more changes, and there's even an extra one in the invasive, and similarly with these others. And in fact, the majority, 10 out of the 13 where we saw ADH and carcinoma in the same breast, the carcinoma had extra events, illustrating that genetic progression associated with the phenotype change. We did the same thing for papillomas, and particularly atypical papillomas seen in the same breast the majority of them were also clonally related. And I really like this case. It's very unusual. So there was an atypical papillary lesion, papillary DCIS, and then also a mucinous invasive cancer. And they share events between all three of them, indicating a clonal relationship, that perhaps these few events here were what kicked it off. But then the mucinous IDC went down a completely different route, where it just has this additional 13q loss. On the other hand, these papillary lesions went a different way and gained extra genetic events, including a HER2 amplification that were not seen in this invasive cancer. So you've got quite an early branch going off there, but again, initiating from that same cell. So mapping out these changes that we saw between the ADH and what they progressed into really helped us to decide on what chromosomes we wanted to look at when we were developing a biomarker for upgrade. So what we really want to know with this is if we take a biopsy from someone and we sample it and it looks like ADH or it's not really clear, if we can look at the copy number changes that might suggest that there are additional cancers nearby, or that we may have even sampled the cancer even if it didn't look like it, that would help us to determine whether there was a risk that there was a cancer nearby that couldn't quite be detected. So as you can see here, the samples that had an upgrade were much more likely to carry some kind of high risk, so ones that were more often seen in cancer compared to ADH, compared to ones that didn't have an upgrade. And Tangina's current work with the Cancer Australia grant is to develop this into a fish assay, as you can see beautifully here, these signals, which she's now testing on a larger cohort of samples. So quite some time ago, it was frightening to realise that this was actually about over 10 years ago. We also did the same thing with DCIS and invasive cancer, where we dissected out the two areas. And these shared a much higher proportion of their genomes compared to the ADH with coexisting. On average, 83% of their genomes had shared copy number change. And there were differences, but they were a bit subtle. So as you can see here, this is an individual case, and they're very similar in just a couple of tiny changes that are different. And we think that's because the clonal DCIS, when they're seen with an invasive, they're almost too far along that pathway towards, and there's always that bit of that worry that maybe what looks like DCIS here, maybe it has some invasive cells that have kind of migrated up that duct and are just pretending to be DCIS. So there's always that concern that you're not really looking at a DCIS. You might be looking at something that's well on the route to being invasive. So we wanted to take a different tack, and this is why we kicked off this paired DCIS recurrent study. So you take the DCIS, you take its later recurrence, and then you compare the two. And we can ask a couple of questions with this. So firstly, we want to understand what makes DCIS invasive, right? So this is the timeline. When you think about progression, this is what's happening. It's accumulating things. And here you have your clinical presentation, the first surgery that removes the DCIS. But sometimes the surgery, there's some cells that escape, and they could grow to become either another DCIS, but half the time they become a cancer recurrence. But if we can compare these two DNA, so in the same person, just separated by time, maybe that tells us something about what it takes to become invasive. But there was a second question we also wanted to address, which was about the independence of that recurrence compared to the initial DCIS. So if you think about it, sometimes the surgery is brilliant. When Bruce Mann does it, of course it's brilliant. But somewhere else in the same breast, maybe there was another lesion that wasn't clinically apparent but was developing nearby. And so when you have this apparent recurrence, like it's in the same breast and it's probably could even be in the same quadrant, it's actually an independent tumour that's had a whole different lineage compared to the one that you first detected. And so we really wanted to know how often this happened. So this was the study that is ongoing at the moment, but making good progress. So we collected primary DCIS that had a recurrence more than 12 months after diagnosis. And that was just to make sure that we were giving it enough time to actually have evolved from quite a small number of cells that maybe had been left behind or not after surgery. From these cohorts here, we collected the samples from. And at the moment, we have a data set of 66 ipsilateral, so within the same breast, and also 5 contralateral, which are kind of like our controls, right? The cells were dissected from the FFPE sections and the DNA was extracted. And here, a particular shout out to Sakshi and Hugo, particularly Sakshi, who has done probably thousands and like literally bucket loads of sections that she has dissected out that DCIS and extracted the DNA. And these were put onto one of 3 platforms. So if we had very low input DNA, we did a low coverage whole genome, which works quite well for that. We started out using a targeted sequencing panel, but more recently we've been doing whole exome sequencing with a new method from this company, Twist, that actually works quite well on low input and degraded DNA that you get from FFPE. And the clonality analysis is currently being done by Tangina in the lab. So how do we determine this clonality? So it's a bit of a balance. So there's a balance in working out whether something's clonal or non-clonal, and we give different weights to different types of evidence. So at one end, if you have an event that is shared between your primary and your recurrence, and it's rare, so it's a specific copy number breakpoint, a structural variant where you've got that sequence, or a rare mutation that you know is somatic, that is really strong indication that it's clonal. It's very unlikely to have happened independently in the same person. Ones that are shared but maybe more frequent. So for example, if someone has a loss of 16q, the whole arm of 16q, that happens in more than 40% of breast cancers, and particularly in ER-positive breast cancer. So if you see that on its own, it's suggestive that they're clonal, but it's not definitive, because maybe that could have happened by chance. With allelic imbalances, you can get the knowing which copy was lost, and so that can give you a bit of extra weight. But also frequent mutations, so things like a hotspot mutation in PEI3 kinase, like the H1047R, is present in a lot. So potentially that could be have happened independently. Now, definitively saying that something is non-clonal is quite difficult, because it effectively looks at things that you can't see, that are not shared. So if there's no shared events at all, or if there are a lot of unique events, so each of the tumours has multiple events that are completely different, that would push you down towards that non-clonal call. So we can also bring in clinicopathological features, which we haven't really done now, but we will probably go back and compare once we've got the genetic data. How does that correlate to things like ER status? Do they have the same ER status? Were they ipsilateral? Were they contralateral? Were they the same grade? And see how that compares with the genetic data. So in terms of actually the methods being used, so we look at the copy number breakpoints and compare those. We compare the point mutations, and there's a couple of algorithms that have been developed to look at copy number and mutations and try and put a sort of statistical measure, taking into account things like frequency in your population, to try and give you a number to say whether something's clonal or not. And we're still trying to work out exactly which of these is the best. So I'll just give you some examples. Right, so here is an example of a DCis. So this is all of the individual dots here are collections of reads that have been called at a particular copy number value. So it's a little bit messy because there's a lot of reads. But effectively, if something's in the middle, it's not changed. If it's increased, it's a gain in copy number, decreased or loss. So this is a primary DCIS, intermediate grade, ER negative, was treated with wide local excision, didn't have any radiotherapy. 4.5 years later, there was an invasive ductal carcinoma, grade 3, ER negative, HER2 negative. And I think you can see just by looking at these that the profiles are very similar. And if we sit down and count them, then indeed there were more than 40 shared events with shared breakpoints, shared allelic imbalances. There were some unique events, which we might expect because it's evolved, but overall, so many more shared events, definitely clonal, no question about it. So here's another case that's maybe not quite so clear. So it's again intermediate DCIS, but ER positive this time. No radiotherapy, no radiotherapy. And recurred after a 7-year interval as an ipsilateral invasive ductal carcinoma that was grade 1. So you can see that they have shared events in the blue. We've got this gain here and another loss here. But unfortunately, that gain of 1Q and loss of 16Q is about the most common pair of events that you can get in an ER-positive breast cancer. So on their own, not actually definitively saying that it's clonal. And we do have some events that are not shared, as indicated by the red boxes, right? So we've got a gain here, for example, in the recurrence. So this is one where we've got 2 shared. The breakpoints are at centromeres, which we can't map exactly, and so we don't know whether it's precisely the same breakpoint. And we've got 3 unique events in the recurrence. So on copy number, equivocal, but fortunately for this one, we had mutation data, and they shared this AKT hotspot mutation that's only present in 3% of invasive cancers. So on this basis, in combination with the copy number, we can say that that's a clonal case after 7 years. So here's another one further down the less certain line. So on copy number, we can see we've got a few events, quite a number of events unique to the recurrence, and really none that are shared with the primary. They've got separate events. So on copy number, we would say that this is non-clonal. There's nothing on the copy number that suggests they're related to each other. And in addition, when we look at the mutations, there were no shared bona fide somatic mutations. So the primary has a PIK3CA mutation, the recurrence an AKT, and these are kind of in the same pathway. So you wouldn't, they may have gone, gone down a similar line, but they're probably not from the same initiating cell. So this would be one that we would call non-clonal. So you've noticed as I've gone through, I've sort of talked about, we did copy number and then we did mutations. So how much did having that mutation data help us resolve cases that we weren't certain about? So unfortunately, we had 5 cases where we didn't have any mutation data, but we were able to confidently call clonality on 4 of those, and there was 1 that was equivocal. With 7 other equivocal cases, we were able to resolve 3 of them to be clonal. The other 4 remained equivocal, so the mutations didn't really help us in this case. And then also we had 3 of ones that were called non-clonal that actually got categorized as clonal based on the presence of mutations that were shared. And this is mostly because maybe the copy number, there wasn't really much in the copy number to say that they were clonal, but they weren't like totally different, but then the mutations were very similar. But some ones that were called non-clonal remained equivocal. And here's a bit of an example. So here you've got a primary with a couple of copy number changes, the recurrence with not really any copy number changes, so there's no copy number shared. So this one was called non-clonal on that basis of the copy number alone. But when we look at the mutation, well, here's this PIK3CA mutation, but it's our common hotspot that maybe is present in like 20% of breast cancers, and the recurrent had an independent GNAS mutation. So for this one, on this basis, we're still not sure. So maybe they were clonal, maybe it kicked off with a PIK3CA mutation, but maybe that PIK3CA mutation was independent. And at this stage, we don't know. And I'll talk about the issues with trying to resolve this sort of thing shortly. But firstly, just to summarize, the rate of recurrences that we see that are not clonal. So with our contralateral cases, so there were 4 invasive and 1 DCis, and all of these were non-clonal. And this is kind of what we were completely expecting. We weren't expecting that after a diagnosis of DCIS that you would have contralateral cases that were clonally related. On the ipsilateral side, so for those that recurred as DCIS, it was 16.1%, as invasive breast cancer, 14.7%. There's one that we're not sure, we've just got to chase up what the recurrence was for that one. And so you can see around 15% of the cases were pretty confidently non-clonal, but we've still got quite a big chunk where it's equivocal. And this, I guess, leads me to my challenges of doing this clonality analysis. So the first one is, of course, data quality. We're working with FFPE tissue, it trashes the DNA and it can make it really hard. And so we have lost several cases due to poor quality data in either the primary or the recurrence. And once you lose one, you lose the pair, which is kind of really unfortunate. You can also get contamination from normal cells, so maybe it's not 100% pure, and that can make interpretation a little bit difficult as well, or there might be heterogeneity there too. The other problem, which is a biggie, is that there's no matching normal DNA. So we don't collect blood or saliva or anything from these women. It's a retrospective study. So it can be hard to determine if variants are germline. So the mutation stuff that I've presented so far has really focused only on those variants that we are pretty sure they are cancer-causing somatic changes. So like AKT1, PIK3CA, P53, those kinds of things that we're like, this is definitely a somatic change. So there are a lot of other events happening, particularly when you get to exome, right? They're picking up a lot of extra passenger events. But at the moment, we don't know if they're germline or if a germline, and really rare, like they could be a private germline mutation. We filter a lot using these huge databases like Nomad and Exact to filter out as many of the germline ones as we can. But if you see something, if it's not in that, you still don't really know. It could be a private variant that someone's catching. So, and this contributes to these having so many of these cases still be equivocal. We might be able to resolve some of these with confirmatory sequence. So for some of the cases, we could probably dissect out a little bit of the stromal that's in the same block as the DCIS and try and confirm whether we can see them in that non-tumor DNA. And that might help us, because if you just, all you need is 1 or 2 of those that are somatic, and you can call clonality. But one of the other problems is, what do you do with cases where there's kind of no events, like the primary doesn't have any copy number changes? And it's not that unusual for DCIS that this happens. Some of them are quite genomically stable. So potentially we could go to the whole genome sequencing, but you still run into that issue of FFPE is going to make it hard to pick stuff up. And you still have the problem with not having normal, matching normal tissue. So I think we could really only do that if we were able to get normal tissue from these cases to try and resolve those. But what does it actually mean? So the reason why we did this is because we were worried about picking up biomarkers. When you look for a biomarker, one of the ways that you can look for it is to look at the tumor cells. You're wanting a tumor intrinsic biomarker, something about that tumor that says you are high risk and you're likely to recur. But when you're doing that, if 15 or 20 or even 25% of your cases are independent recurrences, then looking at that primary tumor is not going to help you find a biomarker to predict that independent recurrence. It's only useful for predicting a clonal recurrence. So, we had big concerns that in doing all this biomarker work, if we didn't have that knowledge of which ones were non-clonal, it might make it difficult to find tumor intrinsic biomarkers. So possibly we can then say, okay, well, maybe tumor intrinsic isn't the way to go, and what we want to be doing is looking at the environment, and that may be very valid. But even so, and then also, can we find anything different about the patients who had a new tumor as opposed to those who had a recurrence of their previous disease? So we did look at this a little bit, but didn't really find anything. So the ER status, and I'll just point out this is not very complete. We've still got some data to collect on the ER status of the primary. This is not really a particularly strong difference there. Maybe a slight hint that ER negatives were more likely to be clonal to their later ones, but yeah, like I said, not significant. And similarly, grade, not a great deal of difference there. One of the other things that we were interested in was in whether they'd had radiotherapy. So we had a small number of cases that had recurred after radiotherapy, and we wondered maybe they were more likely to be non-clonal because radiotherapy should clean up any cells that get left behind. But actually there really wasn't a difference at all. If they'd been treated with radiotherapy, it didn't seem to matter. It didn't seem to change the clonality rate there. So the other thing we were interested in was whether clonality rates varied by time or by type. And I've kind of seen from the previous one that it didn't seem to make too much difference whether the clonality rate was similar between DCIS and invasive. But what about time? So this is the length of time it took from the primary surgery to the tumor to come back. And we thought that maybe as time went on, you might have more non-clonal cases coming through, but actually there was no correlation at all, right? So if we compare our clonal with our non-clonal here, it wasn't significantly different between the length of time it took for that tumor to come back. There was a difference with the type of recurrence in terms of time, but this might be being influenced by maybe there's something strange about these equivocal ones. So this is very preliminary, and we need to do some more work on this to understand if this is the case, and more cases, of course, as well. But yes, it didn't definitely seem to, but it would be interesting, I think, to see if that changed depending on the ER status. So, when you think about ER status and how it changes recurrence patterns in invasive breast cancer, the ER negative ones tend to come back more quickly, whereas the ER positive ones tend to come back more slowly, but we don't quite have enough. We need to fill in the gaps on the ER status to see if we could see a difference there, and whether that was more likely to be non-clonal if you were, ER negative and 7 years out, as opposed to ER positive and 7 years out. So something to work on. So we're also interested in understanding what the differences there were in the copy number changes overall. So this is, again, a percentage plot across the genome, gains in blue, losses in red. So our clonal cases, so these are the changes that we see in those that were clonal. This is just looking at the primaries. A group of non-recurrent cases and a group of non-clonal cases. And you can see that the clonal cases do maybe have slightly more, though we haven't quantified this yet, but certainly do seem to have some regions of the genome that are more common in cases that were clonal compared to cases that were either non-recurrent or non-clonal. And it was actually really quite nice to see this, because we did a previous study where we didn't know about whether they were clonal or non-clonal, our first study of DCIS recurrence, and some of the regions that we're picking up are actually really similar. So, 11, 15, 17, and 20, if I go back, these are also some of the regions that are different between the clonal and the non-recurrent, which was, actually really gratifying to see, and suggest that maybe we're on the right track with our biomarker that we picked up through the study, even though we didn't know the clonal status of those recurrent cases at the time. So what makes DCIS invasive? So I will say, with all of this analysis that I'm presenting is very preliminary, and I've just been looking at it over the past couple of days with everything that Tangina's given me, and we haven't really dug into this yet very much, but I'll just sort of talk a little bit about some preliminary observations. So in invasive recurrences, this was a case that we did quite early. So this is just an individual case, a primary and a recurrence. And we saw some extra copy number changes in the recurrence. But another change that we did see was the presence of a P53 mutation in this recurrence. Like I said, this was one of the first ones that we did. And that was really exciting because P53, definite cancer gene associated with aggressiveness in a lot of different cancer types. Maybe this is how they become invasive. But actually, when we looked at the rest of them, it doesn't really seem to hold up. So we're seeing similar. So this is neither primary nor recurrence having P53 mutation. This is both the tumor primary and the recurrence having a P53 mutation. And you can see that it's pretty similar between whether they have a DCIS recurrence or whether they have an invasive breast cancer recurrence. And we only saw in the end only 2 cases where there was a recurrence, there's a P53 mutation in the recurrence only, so it had developed. There were a couple of cases where we didn't detect we didn't call a P53 mutation in the primary, but when you go back and look at the files, you can see that there are a few reads there that have that. So just in a really tiny percentage of the primary cells, that P53 mutation was already there. And not really different compared to those that were non-clonal, maybe slightly more P53 mutations in the clonal primaries versus the non-recurrent, but still small numbers at the stage. We need to get more of these non-recurrent cases to pick that up. And it could change again with if we can resolve those equivocal cases. So maybe not P53, but it was a good hypothesis. But the other thing that we were interested in was looking at ploidies. So if you can recall back to those karyotypes that I showed, the pretty colored karyotypes, and that difference between that diploid one and the one that was whole genome duplicated. And so this was something we were interested to see because whole genome duplication in multiple cancers is associated with metastasis and general bad news. So here we're looking at equivocal, non-clonal, and clonal. And this is the ploidy difference. So whatever the ploidy was of the primary, has it gone up, in which case it will be positive, or has it gone down, in which case it would be negative in the recurrence. And while it wasn't difference between non-clonal and clonal, where it was different was between if they recurred as an invasive versus if they recurred as a DCIS, as you can see here. So it was much more likely that there would be an increase in ploidy if they recurred as an invasive compared to DCIS. Now, massive caveats with this type of analysis, because in this case, predicting that ploidy status is based on the mutations that you see, the allele frequencies, the copy number, and it's a model because it also depends on how much normal contamination, the purity of the sample, and basically the algorithms kind of just fit all these different models and go for the one that they think is the best, and it doesn't necessarily represent the ground truth. So if we really wanted to nut this out, we would probably need to go and do some kind of orthogonal analysis like a fish, so we can actually look at it in situ and see, oh yes, no, I can definitely see that there's 4 copies on average as opposed to 2 copies on average, and see how that pans out. But it was still pretty interesting to see that even with all those caveats, those kind of analytic caveats shouldn't make a difference here, but what we are seeing is that difference between DCIS and invasive, which is pretty interesting. So just to finish up with future work, so we're still increasing the sample size. We're aiming for 100. I'm not quite sure we're going to get there because of the failures, but we'll see how we go. Filling in the gaps. There's still quite a few gaps in terms of things like ER status and grade and our clinicopath data. And of course, we also want to try and resolve the equivocal cases through some additional validation, trying to find somatic variants that may or may not be conserved between the pairs. We also need to dig deeper on that comparison between those primary and recurrence pairs to see if we can see differences. Are there genetic differences? Maybe it is that maybe there aren't, but we need to look more closely to see. We also want to see whether there are differences between the primaries and can we predict whether they're going to recur as a DCIS or as an invasive. So far, nothing's leapt out of this, but we haven't looked in detail. Tangina is going to be doing a lot more phylogenetic analysis, trying to build more sophisticated models that can integrate both copy number, amploidy, and mutations, and draw those kind of really interesting evolutionary trees. One thing that would be really good to do is also looking at mutation signatures now that we've done exomes, and particularly if they've had radiotherapy. So if you had a primary DCIS and it's been given radiotherapy and it's come back as a clonal tumor, or even it's come back as a non-clonal tumor, can we see that radiation signature in there? I think that would be very interesting to look at. And Tangina is also working up a single cell sequencing, and this might help us resolve that whole genome duplication. Is it present in the primary? Can we see it? And what proportion is it there? What proportion would it be in the recurrent invasive? And can we pattern out those whole genome duplications and where they might occur? So I'll just finish with acknowledging everybody. So everyone in my team, all the people who contributed by, wearing their little fingers down to the bone, dissecting out and extracting DNA. It's very tedious, and I'm very grateful that people are willing to do that. So everyone in my lab, particularly collaborators such as Stephen Fox, Ian Campbell, and Bruce Mann that help us get the samples, bioinformatics team, and everyone that has, like I said, helped get us those samples together. So thank you very much.
Thanks so much, Kylie. That was beautiful. I'll just remind everybody that you can ask your questions by popping them in the Q&A function that's just at the bottom of your Zoom screen, hopefully. And while you're thinking of some great questions, I might start off with a few that I've been thinking about throughout your presentation. I guess one of them, coming towards one of the parts at the end that you were talking about, is it'd be really great to be able to look at a cohort where they're more likely to have, if I'm understanding correctly, new primaries rather than recurrences. So are there like a group of people, I'm thinking of maybe, I don't know, mutation carriers where they're more likely in some cases to have multiple tumors, where you could get a cohort to look at it from the other angle to kind of sort of see what you're dealing with to work out which ones are clonal versus non-clonal?
Yeah, that'd be interesting. I mean, you would predict that people with a germline mutation in BRCA or one of the other genes might be more likely to have independent primaries. Maybe made slightly more complicated because many women will choose to have a bilateral mastectomy if they've had a cancer diagnosis and are known to have a germline mutation. I mean, not everybody, obviously, but yeah, that I could see that as being a difficulty in getting that kind of cohort together. But it'll be interesting. Like, we haven't. There's a couple of variants that I spotted as I was going through where I was thinking, oh, I wonder if that's germline, because it was in a potential germline gene. And yeah, but we would need to validate that. Yeah,
or maybe not in the BRCA1, BRCA2s, but in the ones that have maybe been tested ages ago for BRCA1, BRCA2. They didn't come up, but we still know they've got a strong family history and they've sort of had numerous cancers. I've got a question here from Bruce. What was the time between index lesion and recurrence associated with clonality?
It wasn't. It varied. There didn't seem to be a difference if they were non-clonal versus clonal. The time to recurrence was not different, if that's the question, Bruce, that you're asking. I can't tell you exactly, I can't pick the number out of my head, what the average was, but it wasn't significantly different.
He said, yes, it is.
Okay, yeah.
Were you surprised by that? I would have kind of, like, when you were explaining, I mean, I know you had the ER positives and the ER negatives in there, which may have kind of, not given you enough power, but.
Yeah, I was a bit surprised. I was kind of anticipating that the longer you went out, the more likely you'd have an independent. But yeah, it doesn't seem to be the case. And I can remember, like, one of the first cases that we looked at was a non-clonal, and it was, a year and a half or something, and you just seemed crazy that could be possible.
And do you think with numbers, you might be like, as you add more, you might be able to sort of see that, I don't know, different mutations or different hotspots of change might be what actually is more driving the changes in the distance and time, because they're not as, I don't know, I guess bad.
I guess P53 might be one of those ones, or the ploidy changes. The degree of that degree of genomic instability maybe that drives the ability to adapt to the challenge of having had surgery and or radiotherapy. Yeah.
He's asked another question. What about ER and PR negative? Were they aligned in clonal but not in non-clonal?
So haven't quite looked at that yet, because we've got, like I said, quite a lot of gaps so I haven't specifically gone and look. I was kind of starting to look at it, and then I realized it was about 30% of the cases that didn't have one or the other for either the primary or the recurrence. That information was missing. So it wasn't particularly, I didn't really think it was worth doing just yet. So when we get that data, that's definitely something that will be interesting to look at if it differs, for sure.
I've got another question, firstly from Eva. She said, really interesting presentation, Kylie. Is there scope to perform single-cell RNA-seq to get around the heterogeneity or tumour purity and get a cleaner signature for clonal versus non-clonal? And I guess I've got a question to add on the end of that, which is, how are you going to do it? Can you get fresh samples?
Well, so the beautiful thing is that actually now you can do spatial single-cell RNA-seq on FFPE. So this is this new stuff that 10x are bringing out. And Stephen Fox has already forked out some cash to give this a try. And David's going to be doing this pretty soon, because it's really interesting. Like if we could, map out that the single cell RNA, and if we could do it with the DNA as well. I was onto the reps like, why can't you do both at the same time? Surely it's possible. But anyway, map out that spatial, because then you can get an idea exactly the heterogeneity, but not only that, like where it is too. So particularly interesting for those cases that have areas of microinvasion. Like, can you actually look at those? Are they transcriptionally different? Are they mutationally different in that tiny, that, as it's happening, it's like invasion in action. Can you see those differences there?
Yeah, that'd be awesome. Another question from Jeff Lindemann. Nice talk, Kylie. Are there any gamma radiation studies that have been evaluated, obviously expecting that in most cases the DCIS would have been eradicated?
So I'm not sure I understand the question.
Signatures.
Signatures. So there are not in, now I'm going to get myself in trouble. There are some signatures for radiation. I've seen a couple of papers. I'm just not sure if they've been done on breast cancer or not. I'd have to go back and have a look, but I've definitely seen a few papers where there have been second primaries after an initial diagnosis, and it might be that it was like a leukemia after radiation for a childhood cancer or something like that, where they mapped out, they were able to detect a radiation signature. And of course, there have been in vitro studies as well where they've zapped and then seen how that changes the mutations.
Yeah, and I'm sorry for stuffing up that question, Jeff. So another question, do you have any insights into the papillomas? Can you identify the papillomas that are more likely to have an associated more significant lesion, so maybe a DCIS or invasive cancer?
I think we will be able to. So this is work that's ongoing at the moment. So Tangina's initial study, which was quite a small number of papillomas, did see definite differences in the copy number events, and also the, funnily enough, the absence of a PIK3CA mutation in those that were more likely to have a cancer associated with them. And so that's what the Cancer Australia /MBCF grant will be doing next, is to validate that using an in situ assay. So she's specifically selected loci for papillomas that we think indicate it's more likely to have a cancer nearby, because that's absolutely being able to predict that upgrade would be really helpful.
Yep. And just I guess for the people that aren't in our labs, right? So how many women have B3 lesions? So if you're able to work this out and work out how to upgrade them, how does that sort of feedback and help the patients?
Yeah, absolutely. So there'd probably be about how many per year? I worked this out at one point. Maybe 1,000 or so that might have a diagnosis, but most of those would be resolved one way or the other. But it's still quite a substantial. Oh, Tangina, you're a legend.
You're a legend.
Yeah. Okay, that was good. I pulled that one out.
Got it right.
So about 1,000 women per year that had that kind of diagnosis, and then it would be helpful to have some kind of assay to give additional sort of confidence that you weren't missing something.
Totally. And for those that are listening that maybe can't see the chat, Tangina has also sort of updated us and said that number climbs, with the population changes in the US, to be 100,000 per year. So it's a lot of women that you'll be able to help then if we can work this out.
Yep.
So I'll just see if anybody else has any other questions that they would like to ask Kylie. Not seeing any come through. Well, I'll thank you for a beautifully delivered presentation that was nice and clear, and has got a lot of people very interested, obviously. That brings us to the end of today's Breast Tumour Grand Rounds seminar. And I'll just remind you that tonight we've actually got the Breast Cancer Education Series. The topic is breast reconstruction, and there'll be case presentations from Dr. Dean Trotter and also from Dr. Vendela Groofman. who are from the women's, and along with that, a multidisciplinary panel. So you can register using the QR code that you can see on your screen, or looking back through the emails from VCCC, of course. And we hope you see you there tonight. And if not, we'll see you in 2 weeks' time when we have the next breast tumour grand rounds. Thanks so much.